Philippines are classified into three classes. We have the class 1. These are caves with delicate and fragile structures or formations, recorded threatened species, archaeological and paleontological values, and the conditions inside these caves are considered to be extremely hazardous. That's why allowable activities inside these caves are limited to mapping, photography, and educational and scientific purposes only. We have also the Class 2 caves. These are those caves with areas or portions which are considered hazardous, but also contain sensitive geological, archaeological, historical, cultural, and biological values, and of high quality ecosystems. It may be necessary to close portion of these caves and open only certain sections to experienced cavers or with guided educational tours or visits. The last class of Philippine caves are the Class 3 caves. These are considered caves which are generally safe for or to inexperienced visitors. And there are no known threatened or archaeological, geological, natural history, cultural, and historical values. These caves may be utilized for economic purposes such as guano extraction and even edible nest collections. This is an example of a Class 1 cave. This is the famous Hinatuan Enchanted River in Surigao del Sur. Licup Cave, Samal Island, as an example of a Class 2 cave. This is in Davao del Norte. And the Puerto Princesa Subterranean River in Puerto Princesa, Palawan, as an example of a Class 3 cave. As of 2020, there were about more than 3,000 caves discovered. This figure shows the number of caves discovered per region, most of which are from Region 4B. And from this number, only 20% were classified. Thus, there is a need to determine and update the biological indicators or factors in classifying caves. This is aside from the fauna, arthropods, and microbes and arthropods can also be used as indices for classification of caves in the Philippines. In 2018, we have initiated study on the microbial diversity of guano from bats. Guano are fecal matter from bats and are harvested by the locals as sources of organic fertilizer. Through combined omics approaches like phenomics, genomics, and metagenomics, we were able to determine the bacterial diversity, isolate and characterize potential and pathogenic microorganisms. This figure shows the diversity of bacteria in guano samples that we collected from 2 chambers of the Caballeriza Cave in Pangasinan. And it's interesting to note that the microbial profile of the guano that we collected from the 2 chambers in Caballeriza Cave has very interesting or revealed interesting microbial profiles and comparable with those reported from caves in India and in Finland, with actinobacteria as the dominant bacteria from these guano samples that we have collected. We all know that actinobacteria are groups of microorganisms with, or particularly bacteria, noted for its antibiotic and enzyme production. Through functional profiling or by predicting the functional diversity, we noted that the functional content of the metagenome from marker gene survey proved functional diversity of the guano or the bat guano samples that we have collected. These interesting findings led us to this presentation of our work on microbes in the obscurity of caves in Calabarzon, Philippines. This presentation is a collaboration among microbiologists, computer scientists, and students in UPLB. The study sites for this project is the Calabarzon. So we have identified 5 caves in Calabarzon, 1 in Cavite, Laguna, Batangas, Rizal, and Quezon. Unfortunately, several caves that we have visited in Cavite do not fit the criteria or the requirements for this study. That's why we proceeded to the study or collection of samples from caves in Laguna, which is in Cavinti Underground River and Cave Complex, Cavinti, Laguna, Kamantigue Cave in Lobo, Batangas, Pamitinan Protected Landscape in Rodriguez, Rizal, and 2 caves in Quezon, one of which is the Sungwan Cave in Tayabas, Quezon. We are still to visit another cave in Quezon, which is the Palale Cave. This study or this project is under the accelerated R&D program for capacity building of research and development institutions and industrial competitiveness, under which the Niche Center in the Region for Research and Development, or NICER, is part of this program. So this NICER is known as the Center for Cave Ecosystems Research, or CAVES, which is the first NICER or Niche Center for R&D in Calabarzon. This NICER CAVES is implemented in the UPLB Museum of Natural History and is composed of 4 component projects. The first project is on terrestrial vertebrate and invertebrate fauna. The second project is on the floristics assessment. The third project is on terrestrial arthropod diversity. And the fourth project is on the phenomic, genomic, and metagenomic analysis of microbiome of bat, gut, and guano from the 5 caves mentioned. So the objectives of this study or this project is to conduct comparative microbiome studies in gut of specific or endemic species of bats present in the 5 caves and the guano of bats collected from the 5 sites which are covered by this program. And through the application of phenomic, genomic, and metagenomic approaches, we will be able to know the microbiome as well as the identity and characteristics of selected groups of bacteria and fungi. And we are also creating a database in order for us to curate these data sets that were generated from this project. So these were some of the photos that were taken during our field visits. So the project 4 has 4 component studies. So the study 1 is on the comparative microbiome studies of bats and bat guano present in the 5 caves mentioned. And this is spearheaded by Dr. Ronilo S. Flores together with Mr. or Professor Andrew Montesilio, and with our project development assistant, Mr. Bonnie Datul, which I believe has presented details of his study or of this study 1 during the best paper competition. And the study 2 is on phenomics, genomics of selected functional and pathogenic bacteria and filamentous fungi, being spearheaded or led by Dr. Noel G. Sabino, together with our project development assistant, Mr. Paul Jan Flores. The study 3 is on the establishment of a resource center of cave microorganisms. So they are developing a database to curate the data sets and all the data that will be generated from Study 1 and Study 2. This is being led by Professor Myla Anacleto, Professor Arian Hasildo, and Professor Fermin Lapitan. And the Study 4 is on the development of a biosafety guideline on wildlife biodiversity assessment and capacity building of biosafety and biosecurity protocols and risk assessment, which is being led by yours truly. So the Study 1, just a quick presentation and just a quick presentation of the different studies under this project. We have the comparative microbiome studies of bats and bat guano collected from the 5 caves in Calabarzon. So here we would like to know the microbial community of these samples from different species of bats that were captured inside or outside these caves. So we would also like, aside from the microbial community profile, we would also like to know the functional and potential biotechnological diversity of the important genes through metagenomics using high throughput next generation sequencing. So this is actually the workflow that we have followed for this study. So we have our samples of known species of bats. So we collected the droppings or the fecal sample, and we also sacrificed 7 individuals of bats per species. This is actually allowed through the gratuitous permit that we get from our partner in the Department of Environment and Natural Resources. So we extracted the community DNA, and these were community DNA, and the extracted DNA was used as a template for the amplification of the V3 and V4 regions of the 16S rRNA, and the samples were submitted for sequencing in international and local facilities, and data were then pre-processed, and the sequences were analyzed and compared with those in reputable sequence database in order to determine the taxonomic classification of these sequences. We are also performing functional diversity analysis of these gene clusters. And on the initial, which I believe has also been presented by Mr. Boni Datul in one of the sessions of this convention, we have collected several species of bats from the 4 caves that we have already visited. And common to all these caves are bats of the species Deinocheirus jaguari, and this is endemic in the Philippines. So we have collected at least 5 individuals per cave or per species, collected in the cave. And based on the initial study, we have already completed the profile of the samples that we have collected from Cavinti Underground River and Cave Complex that revealed the abundance of microorganisms belonging to the phyla Firmicutes, Proteobacteria, and Campylobacterota. Next is our second study, or Study 2, which is on the phenomics and genomics of selected functional and pathogenic bacteria and filamentous fungi. So from the title of the study itself, we will know that we are targeting selected bacteria and molds. And through application of polyphasic approaches, we were able to determine the ID, and we're able to characterize these specific or targeted groups of bacteria and fungi. And we are following this process flow for the samples that we have collected, again, from the same bat trappings and guano that we have collected and used in Study 1 for a specific group of specific species of bats. These were samples were transported and brought to the laboratory for isolation and application of phenomic approaches using cultural methods for isolation, purification, and assessment of the isolates, applying genomics to determine the ID of the bacteria and the fungi, phylogenetic analysis, as well as screening. An example that is presented here is for the screening of antibiotic. We will also be screening for the presence of enzyme and for pigment production, particularly for molds. And based on the initial inventory of the isolates collected, we have a number about 35% enterics, followed by 22%, which are putative E. coli. And we also have several isolates of actinomycetes, salmonella, and lactic acid bacteria. And of course, a number, about 7% of molds. This through the help of our interns and students, thesis students from the Institute of Biological Science, Microbiology Division. We were able to assess and analyze all these isolates. So I think two of our thesis students have already presented their initial findings. Ms. Ayesa Angeles and Ms. Kyla Austria also presented during this convention. So for study 3, inspired by the BACDIVE database, we will also be creating our own database to standardize and digitize the bacterial genome data and metadata that will be collected in this study. And hopefully, those who are working on or are interested in working on cave microbiology would like to use this as their reference or perhaps contribute to this database once we open this database to the public. And lastly, we have already produced a manual on standard operating procedures to follow handling of bats and all the samples from the bats and in the caves. And this will be printed and made available to our partner SUCs and LGUs. And hopefully we'll be able to reproduce these and provide to other researchers and students who would like to work on caves and bats. So we will also be having some trainings regarding bat handling, as well as the processing of samples from bats and caves. So all these preliminary findings, and we are still on our third year, and we are still working on a lot of activities to achieve the targets of these projects. So this will not be made possible without the team who are 50% are microbiologists and active members of the Philippine Society for Microbiology. And we also have a career incentive program fellow who work with us also in this project. We are hoping to have more microbiologists to be working with us. Right now, we have 14 microbiology interns working on this project, and we hope that we will be able to partner with other institutions who would like to pursue research on caves and its environs, particularly on the microbiology aspect of cave studies. So this will not be made possible without the collaboration between UPLB and the DOST, or the Department of Science and Technology Science for Change program, and our interns, both from UPLB and non-UPLB universities. Should you wish to discuss with us our initiatives on cave microbiology research, you can work with us. We have right now 5 partner SUCs, one from each region, one from each province of Calabarzon, Cavite State University, Laguna State Polytechnic University, Batangas State University, University of Rizal System, and the South Luzon State University. You can follow us through our social media pages, and you can send us an email through our nicercaves.uplb@up.edu.ph. These are the references used in this presentation, and we hope to be working with you in the near future. Let us work together and let us explore, preserve, and conserve our caves in the Philippines. Thank you very much, and good day to everyone.
Good afternoon, everyone. My name is Lawrence Victor Divito. I'm here to present a paper entitled The Emerging Toxin Producing Chronic Microbes in the 7 Lakes of San Pablo Laguna in the Philippines, along with my co-authors, different institutions, UST Picard, UPLB Los Baños with the Institute of Biological Sciences in the Division of Environmental Biology Division and School of Environmental Science and Management. According to the study by Carbon Brief, the Philippines is at a threshold or a tipping point in climate-relating tipping points, where as you can see that the Philippines is at a vulnerable of less than 2 degrees increase of temperature due to climate change and global warming. Hubs formation may be linked to the increase in temperature and nutrients worldwide, according to a study by Lechman and Murder in 2022. It echoes where, as you can see in the diagrams, that the Philippines is much vulnerable to a 30 degrees Celsius threshold, and also in nutrients for nitrates, where 0 to 10 range of 0 to 10 micromole per kilogram have been predicted in the study. With a combination of temperature and no nitrates, they have predicted that the Philippines is near an extreme 20%. So these temperature and nutrients are very important factors when it comes to hub formation in across the globe. Hubs have been very much well studied and thoroughly studied in worldwide. But in this study, it was disappointing that only Lake Yana was included in the study, where in the Philippines we have a very much high number of lakes in the Philippines. With this regards, hubs in the Philippines, especially in the tropical freshwater, have been overlooked and understudied. So maybe with this conference, we may be able to encourage researchers on this to study hubs formation. The 7 Lakes of San Pablo and focus. 7 Lakes of San Pablo is a critical condition or paralyzed by growth due to number of factors, such as it is a threatened lake. The Laguna 7 lakes have been proclaimed to be the most threatened since because of maybe high vulnerable to anthropogenic inputs, human activities, natural phenomenon of algal hubs or leak overturn, as well as aquaculture practices. It also may be the ecological carrying limits of the lakes, of especially in Lake Sampaloc, where we can see that the limit of the ecological carrying capacity of the lakes have been limited due to the malpractice of certain groups in the fish cages or clustering of the fish cages in the lake. So we need to somehow manage to have a managed sustainability across the 7 lakes of the 7 lakes of San Pablo. This phenomenon, fish kill, is where it is a yearly occurrence in maybe in a certain dry season or wet season, particularly during the cold months in December to January, where as you can see that the fish kill happens when there's an increase of surface water, or maybe an indication of maybe a harmful algal blooms that may be toxic to the fish, or maybe it happens during a leak overturn scenarios where surface waters increased and the lack of oxygen because of a certain population of blooms occur. The objective of the study was to compare the distribution of the phytoplankton community and the toxin-producing microbes based on seasonality in the 7 lakes of Laguna overview of the emerging toxin-producing microbes in relation to the phytoplankton present in the lake, to determine the implication of these toxin-producing microbes in relation to the ecological function of the seven lakes of Laguna. For the methodology, we have conducted sampling in May, November, and August 2022, where we established certain sites, fresh cages, limnetic, littoral vegetation, and littoral households. And using a vertical integrated technique or vertical sampling, we use a modified water sampler and was filtered through a 30 micron mesh size net. And once we have got the sample, we took a 1 liter from each site and preserve it in Google solution and taken is 50 ml subsample were obtained from the 1 liter and settled it overnight and taken an aliquot of approximately 1 ml obtained for identification and counting using a microscope and hemocytometer for quantification of plankton present in the samples and using a online database and taxonomic references for the identification of the identity of the phytoplankton and using data were visualized and analyzed using our software. As you can see, the abundance of the phytoplankton distribution in the 7 lakes, we have sampled during the dry season where Lake Mahicap is abundant of cyanobacteria due to the increase of maybe temperature. And in comparison to the sampling in August where a typhoon hit, when a typhoon hit prior to our sampling, where as you can see an increase of the atoms was abundant in comparison to the dry season. Lake Palakpakin, a very interestingly co-occurrence of green algae along with the cyanobacteria, where most likely the green algae is much dominating than the cyanobacteria. In Lake Palakpakin, an increase of the atoms was found in 2 sites in Lake Palakpakin. 3 sites in Palakpakin, while a low abundance of the atoms were found in limnetic region or limnetic site in Palakpakin. Increase of green algae was found in some palok during the summer season across all 3 sites except for fish cages where cyanobacteria seems to be dominating. Again, the reason for there were no phytoplankton found in Lake Sampaloc during the wet season is due to the typhoon occurrence prior to our sampling, and then an increase of desmids was found in limnetic site. In Lake Bunot, abundance of the atoms was very much dominant during the dry season or May 2022, where only cyanobacteria was dominating a certain fish cage, certain sites like fish cages and littoral vegetation. And Lake Bunot was dominated by cyanobacteria at 3 sites, except for limnetic zone where most of the cyanobacteria is dominating that particular lake. As you can see, that Lake Yambo has been dominated by the green algae, and only on certain sites cyanobacteria seems to be dominating. And again, in August, maybe due to the shift of climate or weather, there's no phytoplankton found in a particular site except for abundances of green algae in 2 sites. And also diatoms are somehow co-occurring with the green algae in 2 sites of Lake Yambo. Interestingly, Lake Panding, even though it's a clear water, has dominated by cyanobacteria. And so surprisingly that this may be an indicator that there's need to be monitoring in the 7 lakes of Laguna, even though they are in a state of in a threshold of the DOA water quality. And again, in Panding, shift of desmids have been found during the wet season, maybe due to the disturbances because of the mixing in the water column at a particular sites in that particular lake in Panding or sites in Panding. Again, highly diverse. Again, it's not surprisingly that Lake Kalibata is dominated by cyanobacteria in the dry season, where most of the cyanobacteria are dominated across all sites. Very interestingly, that Lake Kalibato has a highly diverse phytoplankton group during the wet season, maybe because again the mixing of lakes. So we can establish that there is mixing happening in the lake, and of course due to the weather disturbances. And in certain studies that there's a certain population that may be dominating in each of the depths in the water column. New records of the Lecospermum was found in Lake Calibato during the May season, where it's all found in all sites except for fish cages. The presence of these this Lecospermum was found, but at low population at Sampaloc and Mahicap during dry season and Lake Pandin at August. As you can see, is that the morphological description match the micrograph that is present in the slide. Another new record of cyanobacteria that that have high toxin producing microbes or plankton or phytoplankton is the plankton pigs. This was found in Lake Calibato during May 2022 sampling, and the presence of these cyanobacteria was found in Lake Pandin in the wet season, but at low population, maybe due to the mixing because of certain groups have been dominating in the water column. So maybe this time, a co-occurrence of certain phytoplankton groups that may be present in that particular station. A distinguishable characteristics of plankton is that they have no heterosis, unlike other cyanobacteria that they have heterosis and no affinities. And as you can see in the presence of vacuoles or gas vacuoles are present, these are not your typical plankton because they float. So they have been separate. They float. They float unlike unlike they float because of the vast vacuoles, and they are very sensitive to light and environmental conditions, and they are vulnerable to mixing of the leaks. So we can see another new record is called the Naiadinium, where this is the only freshwater dinoflagellate that can cause fish kill. But still, this is a preliminary identification. Since as you can see is that the morphology is still not clean yet, so we need to determine its morphology. But as you can see in the diagram, there's an unusual intercalation of tabulation of the circulum in the dinoflagellates, and we need further identification needs to be investigated. So, but this is found usually in Lake Calibato again, and also again population of dinosaurs was really present at all stations. Other hubs or photosynthetic microbes that are found in the sampling that we have done during dry and wet, we have found the Cynodospermum, Holococera, Spirulina, Serratium, Primitium parvum, Volvox, Microcystis, and Peridium are the most common cyanobacteria and dinoflagellates. The dinoflagellates that are found in the 7 lakes, while the presence of Spirulina and Cynodospermum is an interesting since because this was occurred during fish kill. And again, even though non-hubs formation, even though they are not toxin producing cyanobacteria, they can still cause fish kill. And this was the case of Holococera. This is the case of Holococera during our sampling in December 2022 to January 2023, where Holococera has dominated certain lakes, but with co-occurrence of other plankton cyanobacteria. So this was the case of a high population, second highly population of a plankton group on the atom. This is Holococera granulata, where it has occurred with several other species of diatoms as well as cyanobacteria. So, it is possible that non-hub species like diatoms can cause also fish kill in the lake. Interestingly, these are found occurring with other phytoplankton groups. In conclusion, dominating phytoplankton shift in relation to season, diatoms dominate during the wet season, or cyanobacteria dominate during the dry season. Seasonal changes may have altered the population of certain groups, and taxon dominated by dry season due to the different environmental factors, and increase of these may be factors connected to the competition, occurrence, and resource efficiency between other phytoplankton taxa or group that may disperse under phytoplankton group. And presence of these emerging phytoplankton are possible indicators of the water quality in the lake is already eutrophicated and leading to degradation. Example, like Calibato. So we need to closely monitor these vulnerable leaks, and also various groups have ecological niche and aquatic trees and have eco-evolutionary changes in time. We need to further investigate the hubs and non-hubs in the 7 lakes, including the assessment of the effects of the water quality and the nutrients, cyclic abundance, and diversity of other aquatic organisms. And we need to further investigate using molecular approaches of the 3 new records that are found. Um, these are maybe present in the lake beforehand, but not yet studied well. So that's why that's why we put it as a new record, since it's it's already been there but not yet studied. So it's been overlooked through time. So this is the a pre-identification of these toxin produced in microbes. And with that, I would like to thank the following people and institutions, and thank you very much for listening.
Aspergillus fumigatus, it is a member of the Filamentous ascomycete genus Aspergillus. It is a saprophytic fungus with a worldwide distribution. These fungus commonly occur in soil and decaying organic materials. They are thermophiles, in which they can grow in high temperatures up to 50 degrees Celsius. They have these specialized strategies to acquire wide range of nutrients, and they are documented to be an effective composting agent to improve soil fertility. Despite being an effective composting agent, Aspergillus fumigatus can cause invasive aspergillosis and chronic pulmonary aspergillosis in immunocompromised patients and with pre-existing lung conditions. A study in European countries from 2009 to 2016 have shown that invasive pulmonary aspergillosis is an emerging clinical entity in intensive care unit patients with respiratory failure. Soon after the pandemic, COVID-19 associated pulmonary aspergillosis has been reported with mortality rate of 52%, including Aspergillus fumigatus in WHO critical priority group in 2022, along with Cryptococcus neoformans, Candida auris, and Candida albicans. Moreover, case studies have shown that environmental isolates of A. fumigatus were genotypically undistinguishable from the strains isolated from invasive aspergillosis patients. In addition, several studies also noted the potential of home environment as source of aspergillus infection among immunocompromised patients. More importantly, the use of azole fungicides in agriculture contributes to the spread of azole-resistant A. fumigatus, making these infections hard to treat and life-threatening. In the Philippines, limited surveillance studies were performed to characterize the environmental and clinical A. fumigatus, and with this, the actual public health burden is not yet fully elucidated, underlying the importance of performing surveillance studies in the country. And with this, together with Dr. Aspergillus fumigatus study entitled Characterizing Clinically Significant Aspergillus fumigatus in Agricultural Environments. This study is significant particularly in the Philippines, where agriculture is a large industry. Therefore, there is a high classes, and in 2020, ang dami pong plantito and plantita, and there is an increasing trend of indoor plant cultivations, and where using commercially sold organic fertilizers are common. Yet, there is limited surveillance studies performed to characterize environmental A-fumigatus isolates. Considering all of these, we have seen the importance of performing surveillance studies on these clinically significant fungus. And in this study, we have this general objective to isolate and characterize Aspergillus fumigatus strains collected from compost piles in vegetable production areas and commercially sold organic fertilizers or CSOF. On this note, we have these three research questions. Are cultural and morphological variations observed between compost and CSOF isolates? Second, are A-fumigatus strains isolated from compost and CSOFs are phylogenetically distinct? Third, does Aspergillus fumigatus isolates exhibit high nucleotide diversity? And to answer these questions, we have performed the following methods. And first is the sample collection and fungal isolation. So we have collected 4 compost samples from lettuce and Chinese cabbage farms located in Cubungan and La Trinidad, Benguet province. Benguet is the largest vegetable producer in the Cordillera Administrative Region. And along with that sample, we also surveyed on the management programs that the farmer used. On the other hand, 3 CSOFs were brought online, and it is important to note that all of the commercially sold organic fertilizers were produced locally. Then we have performed the standard dilution method for isolation, and we cultured the isolates in SDA and incubated them. ~45 degrees Celsius to select fungal growth. And second is the cultural and morphological characterization, in which we have recorded the colony morphology and the microscopic structures of isolates. In this case, selected fungal isolates were cultured in SDA and incubated at 40 degrees Celsius. Then we have observed them for 4 days. After that, we have performed phylogenetic analysis, which includes DNA extraction, amplification of IDS and actin genes, and DNA sequencing, and then the phylogenetic reconstruction. After that, we have performed the DNA sequence diversity analysis using the ITS and actin sequence, and we have analyzed that through DNA-SP. For the results, first question, cultural and morphological variations observed between compost and CSOF isolates. In this study, a total of 14 Aspergillus species were isolated from compost files and CSOFs, shown here in the slide. The upper rows represents the surface of the colonies here, and the lower rows represents the reverse colony photographs. Here, 8 isolates were obtained from CSOFs labeled with A to H, and then 6 isolates were obtained from compost files in Benguet labeled from I to N. Based on my tally, variations in colony morphology were evident among isolates ranging from green, olive green, and blue-green to gray colonies. Considering that the inoculation method used was through inoculating needle, difference in growth patterns were observed. Some extended their mycelia, yet all isolates showed raised, wrinkled, granularly textured colonies with white margins and dull cream to orange undersides. With regard to the microscopic structure of isolates, most of them showed typical Aspergillus fumigatus structures, including branch hypae, short and smooth-walled conidiophores with globose, pyroform, and spatulate vesicles. Conidioforms also have uniseriate and columnar heads with globose conidia extended from phyalides. And going back with the question, are cultural and morphological variation observed between composed and C-soft isolates? The answer is no, as cultural and morphological isolates than between groups. For the second strains isolated from composed and C-softs are phylogenetically distinct? To answer this question, neighbor joining and maximum likelihood tree construction were performed. And we have observed that the neighbor joining and maximum likelihood trees for each gene have the same tree topology. However, in this slide, we only show tree for ITS with figure A and actin in figure B. And we have observed that there are two major clusters. The first cluster, shown with violet arrow, includes 11 local isolates that clustered with Aephemegatus reference sequence. In this cluster, we can observe the isolates coming from compost files and C-softs. In contrast, the second cluster, shown with red arrow, includes isolates which are clustered with Aspergillus nedulans. In the two loci here, only PMO FA F1 2S1 and GQCA F2 2S2 have shown to consistently clustered with Aspergillus nedulans. From this, all isolates from compost files and 6 isolates from C-softs are confirmed to be Aephemegatus, while 2 isolates from C-softs are confirmed as Aspergillus nedulans. From here, are Aephemegatus strains isolated from compost and C-softs are phylogenetically distinct? The answer is no, as Aephemegatus isolates are closely clustered regardless the origin and loci used. For the third question, does Aephemegatus isolate exhibit high nucleotide diversity? In this study, ITS and actin regions were analyzed in the NASP version 6, excluding isolates clustered with Aeph nedulans. From here, we have observed that ITS and actin obtained nucleotide diversity index of 0.01512 for ITS and 0.00135 in actin. And comparing that to the study of Irony 2015, analyzing ITS of Aephemegatus strains coming from various countries, our local isolates showed higher nucleotide diversity index compared to the study of Irony. And actin have showed lower nucleotide diversity index than ITS. From here, does Aephemegatus isolates exhibit nucleotide diversity? The answer is yes, as ITS nucleotide diversity index of Aephemegatus local isolates were relatively higher than the study of Irony et al. In 2015, accounting isolates from various countries. In summary, a total of 14 Aspergillus species were isolated from compost files and C-softs, 8 of which were isolated from compost files in Benguet province, and another 8 were isolated from C-softs. All isolates from compost files and 6 isolates from C-softs are Aephemegatus, while 2 from C-softs are confirmed as Aspergillus nedulans. Cultural and morphological variations were observed across isolates than between groups. Aephemegatus isolates are closely clustered regardless of the origin and loci used. Lastly, Aephemegatus local isolates showed high nucleotide diversity for both loci, which includes the IDS and actin. In conclusion, a close phylogenetic relationship between Aphiomegatus isolates, regardless of the origin and loci, suggests that Aphiomegatus isolates from compost and CSOFs are similar, indicating precautions in handling CSOFs for indoor plant cultivation with potential hazards in high-risk individuals. Before the end of my presentation, we would like to extend our thankfulness to the University of the Philippines Enhanced Creative Work and Research Grant under the Office of the Vice President for Academic Affairs for their financial support. So this is the end of my presentation. Thank you. Aspergillus may
Thank you to all our speakers. We now move to the open forum. Dr. Pierangeli Vital, Division Representative for Aquatic Microbiology, will moderate this session's open forum.
And Ms. Fatima Balata to join me on stage for the open forum session. Please feel cozy on your couch. Alright. So for those of you who are here with us in person, kindly tell me to raise your hand. So our technical team member will approach you and please stand up and please state your name and affiliation if you have questions. We can now open the floor for questions for Dr. De Leon, for Mr. Vitu, and for Ms. Balata. First question, please. Right. I think you have one microphone in front. You can approach the front if you have a question. Yes, sir, please come in front so you can be heard. Also, for those participants in our online platform, please key in your questions so our technical team members will read your questions later. Go ahead, sir.
Good afternoon. I am Mike, a microbiologist practicing in industry. My question is for Dr. Marian Bautista and for the team.
Dr. De Leon.
Dr. De Leon. Sorry.
Ang dami ng name, ma'am. Ako din, may coin one word for ma'am. 188, uso daw ngayon sa mga scientists. Dr. Marian Bat Woman De Leon. Go ahead, sir.
Sorry, Dr. Marian. Again, I would like to commend your team for exploring those type of environment for the study. My question would actually be on the methodology. I was able to attend with the presentation of Bonnie, and my question is regarding on the bias when it comes to the sampling. Particularly, it was mentioned that the whole intestines or gut as being used for the extraction. How did you ensure that the sample population or the microbial community is being properly represented, particularly in the poor population or microbial population from that?
We have already optimized the conditions in preparing the sample for the analysis. We have several setbacks, but during the optimization, we were able to correct or improve the methodology. You're right because we just ensure that there is a very higher yield in the extracted DNA, and we have a good quality DNA that we submitted for the sequencing.
A follow-up question po, would you consider po in the study to segment the gut for the sake that we could thoroughly understand the microbiome and its role for the say nutrient and adaptation of the host, and also to properly to properly represent the microbiome?
Perhaps Bon can answer this question if we can do the segmentation because we have only how long is the gap? How many centimeters only is the gap? If we're going to do the segmentation, it might not give us enough sample to be processed. So that's why we have to get the whole gap to ensure that we have enough sample for the sequencing requirement. I hope I was able to answer your question, but I think the best person to answer that is the person who is working with that, which I would like to recognize is Mr. Bond Gatil.
Thank you very much,
Thank
Dr.
you.
Martin.
Thank you for the question. You want to add something? Maybe you can acknowledge and call the co-author your insight? O, di ba, Bon? Sabi ko sa'yo, mag-ready kayo. Oo. So, would you like to add something, Bon? Go ahead, sir.
Good afternoon, everyone, and thank you for the interest with our study. So we, based on literatures, there were researchers who actually studied the gut as well previously. Some of them did segmentation, so they cut the gut into 3 sections. The problem with our study, we are only trying to look at the totality, so for the whole intestine. And since the objective is to really just it's not in the scope of the study to compare the microbiome content at the different segmentation of the gut. Hence, we didn't do it, and we only we extracted the total DNA from the totality of the intestine. So and we composite the samples prior to actual total DNA extraction. Thank you.
Thank you for the answer. Also, thank you, Dr. Julian. Another question from the floor. Dr. Celine.
Magandang hapon po sa ating lahat. This is for the second speaker. I think it's Mr. Vito. Okay. Now, you mentioned that you were able to observe the presence of 3 new toxigenic cyanobacteria from the 7 lakes. Okay. So the first question is, since you mentioned that these are toxigenic, can you please inform us what are the toxins which are being produced by these organisms? And second, were you able to determine the trophic state index of the different lakes? Because definitely this will have an impact on the so-called diversity as well as presence of organisms or this phytoplankton in the 7 lakes. Thank you.
Thank you, sir. So for the first question, actually, we will not be looking for the toxins for the cyanobacteria since our project is really on the water quality assessment of the 7 lakes of San Pablo and also the abundance of phytoplankton. For your second question, in relation to the trophic states, we're currently doing a modeling for the 7 lakes of San Pablo, which is part of the preferred funded project. So definitely there will be new information regarding this in the future since we preliminary investigation of the project. But thank you very much for your answer. Per question, sorry.
Any follow-up, sir? Go ahead. Freshwater cyanobacteria usually produce anatoxin, multicysteines, and these can easily be detected. So there are different methods. Meron nga yung traditional methods that you can use to detect these toxins, and they can easily be done in the laboratory. Maganda siguro that is you want to confirm if they are really toxigenic kasi there are cyanobacteria whose, sabi natin, depending on the species, some are toxigenic, some are not. So you mentioned that these are toxin producing organisms. So siguro at this stage, huwag muna po natin siguro muna sabihin na these are toxigenic cyanobacteria unless we have really proven that they are toxigenic kasi you have to consider what are the species present in the said body of water before we can say. Siguro, a recommendation ko lang is we can do identification, and then from there, we can in a way predict if it is toxic and or toxicity, and also to detect for the presence of toxin. Thank you.
Yes, sir. Dr. Signo, I wanted just to clarify, we will not be doing the toxin on the toxin producing culture. But this is based on previous studies by in other countries that these are specifically those three species are toxin producing. Though we will not dwell on more details, but these are literatures. These are found to be toxic on other countries. So this would be somehow inferred in the Philippines. These phytoplankton are not well studied because of previous studies in the Philippines, but they are needs to be more studied conclusively and comprehensively. In fact, this would be a preliminary study again for the future project, but it would be nice to have a collaboration with you for further investigation of these toxin producing microbes. Thank you very much.
Thank you for okay. Alright, we'll move on to the next. I see hands here. Please approach for our microphone. And for those of you at the back, if you have questions, please go to this part of the floor.
Good afternoon, I'm Rhonda Fons from IDEA. First, I'm not going to ask my co-author question, but I'm going to ask Dr. Warren De Leon, particularly on fungal, because of what we've funded. So, when you mentioned that you were interested in working or looking into, pathogenic bacteria and molds, were you looking at pathogenic fungi, pathogenic to the bats, or
Actually, no, we are not looking at the pathogenic fungi, but more of beneficial fungi, because we are targeting fungi that may produce antibiotics or enzymes or pigments. So yun muna yung target, because we are already working on the microbiome, and mostly are pathogenic. So para there is balance, we work with pathogens, we also work with beneficial microbes.
because I was interested on bats that or like a fungal pathogen of bats with Bacterium dendropatitis, which is causing the white nose syndrome in many bats all across the globe. So I'm wondering if it's already present in the country.
Fortunately, based on our previous capture of bats, we have not encountered yet this type of fungal pathogens. Hopefully, we will not be able, because this is another biosafety concern.
Thank you very much.
I think we are opening up collaborations for everyone. Okay, so can I entertain more questions? We have time left. Any other questions from the floor? Please go to the side. I have a question for Ms. Balado. I'm waiting for our participants to come on stage. With all of this platform, so what are you thinking of any applications of your isolates right now? With regards to applications, we don't really focus on that, but we wanted to characterize, and thank you for your question. Okay, so we wanted to characterize more of these isolates, particularly on their phenotypes and the genes that are related mostly in their mating. The mating is a potential in the virus. That's what we wanted to unravel also. So now, if you are able to identify certain traits of our isolates, we can suggest and recommend or identify the public health threat in the Philippines, as it was not really yet established or identified in the Philippines. Good luck po and congratulations. I believe there are few mycologists in the country or those working in your project or organism. So yeah, congratulations. So, I think meron pa po bang questions? And if go ahead po, please approach the microphone, please state your name and affiliation.
Hi po, from University of the Philippines. And this is for Dr. De Leon.
Sorry, sir.
Have you identified microbes that have such that they are better to function together.
Okay na po. Saka po.
So far, we have not yet considered that because based on the objectives, we just want to focus on the objectives of the study. But that is a good suggestion that the team can look into, perhaps on the phase 2 of the project. So if someone would also like to work with us, just like what Dr. Vita said a while ago, that we can always collaborate. Like if you mentioned that you're working with or interested with this fungi, kung may makikita kami, sana wala, we can always collaborate. So thank you very much for that question. But right now, wala akong masagot sa kanya because we have not yet considered that in this part of the project.
Maybe Mr. Malababa can do that for his graduate studies perhaps. Okay. Any questions from our online participants? Alright, any other questions here
Opo, sir.
on the floor?
Pero kahit isa lang dun, sir, okay na daw.
So we wrap up the first part of our technical session. So
Mobile na, sir.
thank you all for your engaging
Sure,
questions and active participation. So please stay on stage for our presenters, because I would like to call on stage Dr. Howard, our PSN president, and Ms. Hannah Jane, Mr. Emerson Cruz, our technical paper chair, to award the certificates of appreciation to our first 3 presenters for our technical paper session. Please, sir. All right, so allow me to read the citation first for Dr. De Leon and for co-authors. The Philippine Society for Microbiology, Inc. awards this certificate of appreciation to Dr. Marian De Leon with her co-authors PJF Flores, BB Bull, RJB Flores, and AB Montesino, and NG Sabino for presenting the lead paper for technical session B entitled Out There in the Dark, Microbes Within the Caves in Calabarzon, Philippines, during the 52nd Annual Scientific Convention and Scientific Meeting of the Philippine Society for Microbiology, Inc. in cooperation with Trinity University of Asia, with the theme Microbiology for Unity and Prosperity through collaborations and industry linkages, held at the Hilton Manila via hybrid from July 20 to 21, 2023, given this 21st day of July 2023. Signed, Emerson Cruz, the chair of the Technical Session Committee, and Dr. Howell Hall, the PSM president. We also call on stage if the co-authors are here to join our presenter, Dr. De Leon, on stage for the photo opportunity.
Iba po yun, sir. Pa-out po yun. Oo nga. Hindi niya mag-take yung ano. Hindi rin po mag-take yung mic. Yung speaker, sir, meron. Oo nga, yung mic wala. Pang-out lang ata. Kaya, out lang yan.
So the same certificate is awarded to Alright. The same certificate is awarded to Lawrence Victor D.V.2 and his co-authors J.V.R. Plato, L.E. Cui, E.P. Herrera Jr., R.J.D. Flores, R.T. Benindela, J.G. Kamal F., J.M.D. Balatibat, and B.M. Macandog for presenting the paper for technical session entitled Emergence of New Records of Toxin-Producing Planktonic Microbes in the 7 Maar Lakes of San Pablo. Any co-authors on the floor right now? All right. Okay, and the same certificate is awarded to Katrina May D. Balaga and Alice Bacones for presenting the paper technical session B titled Characterizing Clinically Significant Aspergillus fumigatoris in Agricultural Environments in the Philippines. The same citation and the same signatories.
Congratulations and a round of applause, please. Okay, another round of applause for our first three presenters for technical session B. Now it is time again to introduce our second set of presenters for this technical session. So please join me in welcoming our esteemed speakers who will be sharing their expertise and insights in this session.
Out lang ito.
Let's have the next set of presenters. Paper number 4 will be presented by Miss Mary Ann Shelo V. Relucio San Diego from the Natural Sciences Research Institute and Institute of Biology at University of the Philippines, Diliman. Her paper is entitled Metagenome Assembled Genome of Rayranelia species of a poultry wastewater treatment plant reveals potentially high biosynthetic capabilities. This paper is co-authored by Paul Christian T. Gloria and Mary Christine M. Obusan. Last but not the least, paper number 5 will be presented by Mr. Xander Carl N. Herrera from the Department of Chemical Engineering, College of Engineering and Agro-Industrial Technology, and Microbiology Division, Institute of Biological Sciences, College of Arts and Sciences at the University of the Philippines, Los Baños. His paper is entitled Biomass and PUFA Production of Trostochytrids Isolated from the Selected Mangrove Forest in Batangas and Quezon provinces. This paper is co-authored by MBJ Catli, CMB Masakupan, VP Migo, and LC Villegas. Let us now listen to the presentations.
Good day. The Philippines, being an archipelago, is known for its abundance in water resources. We have beaches, rivers, waterfalls, and lakes, to name a few. We all wish to continue to protect these resources from all the pollutants we create. And one of the facilities that manage to protect these water systems are wastewater treatment plants. They are considered as barriers to hold and remove the pollutants through biologic processes before releasing the treated water back into the environment. There are different types of wastewater treatment plants, but the most common are the conventional treatment plants. And they have this setup. So mostly, the large pollutants are being removed in the primary step. Then the wastewater enters the aeration tank. Then the secondary clarifier. Then the wastewater is treated with chlorine before you release the effluent back into the receiving waters. So if you notice, there is this small setup here, and this setup is mostly responsible for 90% of the wastewater treatment. And this is called the activated sludge. Activated sludge is actually a microorganism drives the treatment process of the wastewater treatment plant. And we know the biological processes happens when the nutrient levels on the wastewater becomes acceptable in accordance with the guidelines of effluent water of a country or a specific industry. However, Most treatment plants do not have any idea on the identity of these microorganisms. Furthermore, there are reports that isolates from these sources have different potentials, such as production of polyhydroxyalkanoates, which are important in the medical and pharmaceutical fields. And then there are isolates that produces enzymes and surfactants, and other isolates with other bioremediation potential, such as metal and plastic degradation. So it would be useful to identify some of these microorganisms and look into their potentials. Hence, the title of my talk is Metagenome Assembled Genome of Rayronella species from the activated sludge of a poultry wastewater treatment plant reveals potentially high biosynthetic capabilities. So first, what are MAGs, or metagenome assembled genomes? So let's break it down word by word. Metagenome is the sequencing of all the extracted genetic information from an environmental sample. So your sample could be soil, water, or the gut microbiota. Some bioinformatic tools can determine which of these sequences or conteins belong together. So they create an assembly for this group of genes, then they create a genome for a single taxon. So these genomes are called metagenome assembled genomes, or MAGs. So when you do your functional analysis, you can now attribute a specific activity to a specific microorganism. For the methodology, the sampling was done on the aeration tank of a poultry wastewater treatment facility to get the activated sludge. DNA was extracted using QIAGEN Power Soil Pro, and the extracted DNA was sent to a sequencing facility. Sequencing was done using an Illumina HiSeq X10 platform, 150 BP pair-ended setting from Nextera XT prepared libraries with a throughput of 15 GB. Bioinformatic tools were used for quality training, and Metaspades was the chosen optimal assembler out of all the other tested assemblers. The MAGs were generated using 3 binning tools and was refined using DAS tools. Downstream analysis included the utilization of Genome Taxonomy Database, PROCA, RAST, PRISM, Anti-Smash, and DeepPGC. After sequencing and performing assembly and binning, the MAG generated was identified as Rayronella species. It belonged to the phylum type Pseudomonadota, class Alpha Proteobacteria, order Hyphomicrobialis, and family Rayronellaceae. It is known to be associated with amoeba and has been reported to be isolated in freshwater, soil, and bamboo litter. So we consider this as a high-quality MAG because it met the high-quality criteria of more than 90% completeness and less than 5% contamination. We also analyzed the reads of the MAG for functionality with regards to its activity as a member of the activated sludge. So these are the genes involved in the nitrogen cycle in the database of FAMA. And we can see that these genes involved in the nitrification were detected in higher abundance compared to the other genes. Now, we compared the findings that we got with an online database called Microbial Database for Activated Sludge, or MIDAS. MIDAS is a resource of information that collects metabarcoding data from wastewater treatment plants around the globe and analyzes their probable functions. In the database, Rayronella was detected in pre-denitrification tanks from samples coming in from the Philippines. So these suggest that the microbe from the poultry treatment plant could potentially have the same activity as the ones detected from the other plants. We have also done general annotation using ProCA, and the genome revealed to have more than 5,000 protein coding regions and 46 RNAs. So the organism can putatively create many kinds of proteins. So we also use another tool for general annotation called RASP, and it has showed us that many of the genes are grouped on these subsystems: the cofactors, vitamins, prosthetic groups, pigments, nucleosides and nucleotides, protein metabolism, fatty acids, lipids and isoprenoids, amino acids and derivatives, and carbohydrates. If we are look more deeply into the activities that are related in the removal of nutrients related to wastewater, these are some of those genes. So nitrogen, phosphorus, and sulfur are some of the parameters that we need to monitor in the effluent. So there are genes related in the nitrogen metabolism for denitrification and for ammonification. And these are the genes for phosphate metabolism, and this is for sulfur metabolism. Aside from looking at the genes, we also look into the biosynthetic gene clusters. So we use anti-smash and PRISM for it and DeepBGC. For anti-smash, we detected 5 unique BGCs, and for PRISM, we have detected 4. So this picture is from anti-smash, where it detected a cluster for the synthesis of beta-lactone and a hybrid BGC that synthesizes a polyketide named rubiginone and a non-ribosomal peptide synthetase. So these two, beta-lactone and rubiginone, are considered industrially important secondary metabolites. Beta-lactone is actually a known inhibitor of homoserine transacetylase, and this enzyme, the HTA, is being targeted by novel antimicrobial agents for its role in microbial amino acid biosynthesis. Rubiginone, on the other hand, is a known antibiotic with a reported activity of increasing the potency of a chemotherapeutic drug called vincristine. Using a deep learning tool for the detection of BGCs, DeepBGC has even detected about 100 more BGCs. So BGC predicts by 2 ways. It's either by class and by prediction. So if you can see the prediction by class, most BGCs have not been classified to any of the following classes. And if you look at the prediction by activity, most BGCs are classified with an antibacterial activity, but there are 26 more that were not classified. So with this, we can see that there is so much more we can explore by identifying these unclassified BGCs, for they may bring about novel metabolites that could be beneficial to our use. So in conclusion, the metagenome assembled genome of Rayronella species revealed its potential activities on denitrification and ammonification as part of the activated sludge microbiome. Also, it could be a potential source of interesting secondary metabolites since we detected beta-lactone and rubiginone. So ultimately, the utilization of MADS is useful in finding novel taxa in an environment and in genome-guided investigation of secondary metabolites. I would like to thank the Microbiological Research and Services Laboratory of the Natural Sciences Research Institute and the Microbial Ecology of Terrestrial and Aquatic Systems Laboratory of the Institute of Biology, UP Diliman. I would also like to thank the UP system for funding this project through the Enhanced Creative Work and Research Grant. Thank you. Good day. The Philippines, being an archipelago, is known for its abundance in water resources. We have beaches, rivers, waterfalls, and lakes to name a few. We all wish to resources.
A pleasant day, everyone. Today, I will be presenting our paper titled Biomass and PUFA Production of Trostochithrids Isolated from Selected Mangrove Forests in Batangas and Quezon Provinces. This will be the flow of my presentation. First is the problem under investigation, which includes a brief introduction about Trostochithrids and their biotechnological applications and the objectives of our study. This will be followed by the methods that were performed from isolation to cultivation to be able to achieve the objectives. Then I will discuss the significant results and findings, and finally the conclusions and some recommendations. What are Trostochithrids? Trostochithrids are heterotrophic estuarine microorganisms which are classified as protists. They share similar characteristics with other organisms under the class Labyrinthulomycetes and family Trostochithriaceae, such as the presence of biflagellates, zoospores, and ectoplasmic. Although they are informally considered as microalgae, Trostochithrids are not photosynthetic, but they derive their energy through saprotrophic nutrition. Trostochithrids are commonly found in brackish waters of marine and estuarine environments, usually feeding on decaying mangrove leaves. They are known to be oleaginous, meaning they can produce large amounts of oil through synthesis and accumulation of high polyunsaturated fatty acids or PUFA amounts in their biomass, particularly DHA or docosahexaenoic acid of the omega-3 fatty acids found in fish oil during their cultivation. Trostochithrids have gained attention from the scientific community due to their ability to produce polyunsaturated fatty acids or PUFA. PUFAs are a type of fatty acid that have multiple double bonds in their carbon chain. They are essential nutrients for many organisms, including humans, as they play vital roles in various biological processes. Overall, the significance of PUFA production by Trostochithrids lies in their potential to provide a sustainable and valuable source of omega-3 fatty acids such as EPA and DHA for human nutrition, aquaculture, and biotechnological applications. Now, wastewater can contain various organic compounds and nutrients which may support the growth of Trostochithrids. To date, there is limited research specifically focused on Trostochithrid cultivation in wastewater. However, some studies have explored cultivation of Trostochithrids using different organic waste substrates, such as agricultural waste, food waste, and wastewater-derived organic matter. Hence, this study was conducted to investigate if it is possible for these protists to be cultivated and propagated in raw and sterile wastewater, specifically distillery wastewater. The general objective of this study was to investigate the biomass and PUFA production of Trostochithrid isolates obtained from the mangrove forests in Batangas and Quezon provinces. The specific objectives include the following. Number one, to isolate Trostochithrids from mangrove forests in Batangas and Quezon provinces. Number two, to characterize the Trostochithrid isolates based on their cultural and morphological traits, road behavior, and 18S rDNA sequences. Number three, to optimize the pH and salinity conditions of culture media using response surface methodology or RSM. And number four, cultivate the isolate with fastest growth and highest biomass and crude extract yield to unsterile distillery wastewater. We go on with the methodology of the study. Fallen senescent mangrove leaves were collected in 2 provinces in Calabarzon. First in San Juan, Batangas, and second in Pagbilao, Quezon. Sample collection was done in 3 different sites per province. The leaf samples were processed in the lab by adapting the methods of Liaño, Arafiles, and Perez with modifications. Cutout leaves were placed on top of the glucose yeast extract peptone seawater agar medium supplemented with ampicillin, streptomycin, and nystatin. Then 1 ml of sterile 100% natural seawater was added on top of the leaf. The plates were incubated at 30 degrees Celsius for 2 to 5 days or until colonies can be observed growing around the leaves. These colonies were picked up and successively streaked on GYPSA medium until pure cultures were obtained. The putative Trostochytrid strains were then characterized based on their cultural and morphological traits. Pure isolates were sent to UPV, Philippine Genome Center for DNA extraction, 18S rDNA, PCR amplification, and sequencing. The growth behaviors of the Trostochytrid isolates were determined by individually growing them in sterile GYPS broth for 9 days. The growth was measured turbidimetrically using spectrophotometer at 600 nanometer. The OD readings were plotted against incubation time to construct the growth curve. Separate culture setup was performed to measure the biomass production. Cultures were grown in 500 ml flasks containing modified GYPSA medium under a 150 RPM shaking condition for 4 days. The biomass was harvested through centrifugation method at 3,500 RPM for 10 minutes. The washed cell samples were freeze-dried and weighed. Biomass production was estimated as gram of dry weight biomass per liter of growth medium. The resulting biomass were sent to biotech for crude fat extraction following AOAC 2003.05 21st edition method of analysis. The crude fat extract was then analyzed for presence of EPA and DHA through fatty acid methyl ester analysis or FAME using gas chromatographic method. Trostochytrid strains were subjected to various conditions of pH and salinity to observe for growth tolerance. pH range considered was pH 1 to 7 and salinity range was 20 to 80 PSU. The setup also determined the optimized pH and salinity conditions that produce the highest biomass using RSM, the software by Design Expert version 11 by StatEase. For the results and discussion, 4 isolates were obtained. Number 1, from San Juan, Batangas, designated as SJB1 ACBL1, and 3 isolates from Pagbilao, Quezon, designated as PQ1 RMB1, PQ2 RAYD1, and PQ3 SAB1. As seen in this slide, all 4 isolates displayed creamy white colonies with rounded shape. The 4 isolates also share the same abundance, opacity, form, and elevation. On the other hand, SJB1 has smooth surface, PQ1 has shiny surface, while both PQ2 and PQ3 have dull surfaces. The colony margin is also different per isolate. Both SJB1 and PQ2 have a rose margin, while both PQ1 and PQ3 exhibited entire margin. The appearance or morphology of the cells under the microscope are also shown here. Here we can see that SJB1 was isolated from mangrove species Aegiceras corniculatum, PQ1 from Rhizopora mupronata, PQ2 from Rhizopora apiculata, and PQ3 from Soneracia alba. The stages of leaf decomposition was also recorded based on their color. The unstained cellular features were observed under light microscope with 1,000 times magnification. Wet mount showed all 4 isolates have spherical amoeboid cell and globose vegetative cell. Presence of binary cell division was also observed for all 4 isolates. They have thick cell walls and their ectoplasmic network differ from well-developed to underdeveloped. After cultural and morphological characterization, their identity was confirmed through molecular identification by 18S rDNA sequencing. With the help of UP Visayas Philippine Genome Center, 2 methods were used for the creation of molecular phylogenetic tree of Trostochytrid isolates compared with other related taxa, the one using the neighbor joining method and the other using maximum likelihood method at 1,000 bootstraps. Although the neighbor joining method can make quick trees, they are not the most reliable, especially when dealing with deeper divergence times. The maximum likelihood method can apply a model of sequence evolution and is ideal for building a phylogeny using sequence data. Either which, our phylogenetic analysis show that on both methods, all 4 isolates belong to the genus Aurantiochytrium sp. with 99.82% identity from blast hits. The growth behavior experiments showed that all 4 Trostochytrid isolates initiated growth within the 24 hours of incubation. Log phase was observed until 96 hours. All 4 isolates attained the maximum growth within 96 to 120 hours until stationary phase. Among the strains, PQ2 showed the highest growth with OD value of 2.93, and PQ1 showed the least growth with OD value of 2.82. In sterile modified GYPS media, the biomass production varied from 9.85 grams per liter to 11.19 grams per liter of dry cell weight. Maximum biomass of 11.19 grams per liter was produced by PQ2, while the minimum biomass of 9.85 grams per liter was produced by PQ3. The total lipid content varied from 24.14% to 45.93%. SJB1 was found to be the highest lipid producer at 45.93%. In our experiments, PUFAS DHA and EPA were not measured quantitatively. Presence of these omega-3 fatty acids was detected in all 4 trostochitrid isolates using the gas chromatography mass spectroscopy, or GCMS. For the optimization of pH and salinity conditions, Figures 7 to 10 showed the 3D surface plot for the interaction between pH and salinity with biomass yield as the response. Details of the analysis of variance table are also presented, and results consistently show that all p-values of the 2 factors considered, pH and salinity, is less than 5% alpha. As a review, when p-value is less than the alpha, then the factor is considered significant. We can visualize these results in 3D plots generated by the ScienXpert version 11. Minimal growth can be observed from pH 1 to 3 regardless of the salinity value, while moderate to good growth can be observed from pH 4 to 7. Now, to summarize, the optimum pH of sterile culture conditions range from pH 5.8 to 6.2, while the optimum salinity range from 20.2 to 29 PSU to support the growth of trostochitrid isolates that will produce the highest biomass yield. Results also suggest that isolate SJB1 at optimum pH and salinity conditions will yield the highest biomass at 15.7 grams per liter among the 4 isolates. These can be confirmed in further studies by confirmatory runs using the selected desirability values. Now, since SJB1 exhibited the highest percent total lipid weight per weight in sterile culture medium and is predicted to produce the highest biomass in optimized pH and salinity conditions, then it was the selected isolate grown on unsterile raw distillery wastewater. Figure 11 shows the setup where SJB1 was cultivated in raw mill wastewater and raw distillery wastewater. After 4 days incubation at 30 degrees Celsius, microscopic observation showed that isolate SJB1 thrived in the unsterile medium, as seen in these micrographs, wherein trostochitrid cells were stained with methylene blue. For the conclusion, number 1, 4 trostochitrid isolates were obtained from mangrove forests in San Juan, Batangas, and Pagbilao, Quezon, and their cultural and morphological characteristics were described. Number 2, phylogenetic analysis of the 18SR DNA sequences revealed that all 4 isolates belong to genus Aurantiophytrium. Number 3, growth behavior determined. All strains attained maximum growth within 4 to 5 days of incubation at 30 degrees Celsius. Number 4, isolate PQ2 was found to produce the highest biomass 4 days after inoculation in sterile culture medium. Number 5, isolate SJB1 produced the highest percent total lipid weight per weight in sterile culture medium. Number 6, all trostochitrid strains tolerated a wide range of pH and salinity conditions. The optimized conditions that is predicted to yield the highest biomass range from pH 5.8 to 6.2 and salinity 20.2 to 29 PSU, with isolate SJB1 predicted to produce the highest biomass among the 4 isolates at 15.7 grams per liter. And number 7, isolate SJB1 was selected for cultivation using raw distillery wastewater as propagation medium. Microscopic observation showed that isolate SJB1 thrived in the unsterile medium. For the recommendation, further studies are needed to determine significant biomass yield when using pH 3 or below and salinity of 30 PSU and above. Next, additional optimization response, specifically the crude fat yield or the total lipid content, can be included in determining the best isolate for cultivation in both sterile medium and unsterile raw wastewater. And finally, omega-3 fatty acids EPA and DHA should be quantified since these are the essential fatty acids with biotechnological applications. Relying only on the crude fat extract analysis will only show the total lipid content of Trostochitrid strains. Here are the relevant references included in this presentation. Our study will not be possible without the support of the following institutions. A heartfelt gratitude to DOST Picard Inland Aquatic Resources Research Division for funding our project. Many thanks also to UP Visayas Philippine Genome Center headed by Dr. Noel Ferrios for the sequencing services of our Trostochitrid isolates. To Department of Chemical Engineering and to the Microbiology Division of IBS UPLB for housing the experiments needed for the study. And finally, to eAsia Joint Research Program for the meaningful research collaboration together with Japan and Indonesia. Thank you for listening.
Thank you to our two speakers. At this point, I would like to call on stage our speakers for the second half of the session, Mr. Yusuf San Diego and Mr. Herrera, to join me on stage for the open forum session. Again, we will have the same process. If you can raise your hand and approach the microphone here in front. Please state your name and affiliation. Okay, let's wait for them to settle down on stage. All right, the microphone is also there. All right, so let's have our first question. Yes, sir, please approach the microphone, state your name and affiliation, please.
Good afternoon, everyone. I'm Edward Manlapas, and I'm from Biotech PPLB. And my question is for Mr. Herrera. Just a curious question. Have you tried conducting validation run on the predicted biomass feed? No.
Please go ahead.
Again, sorry. Have you tried conducting validation runs on the predicted biomass feed? Thank you for your question, sir. So, even though it was not presented in my presentation, but we have already validated the pH and salinity tolerances of the two of our four isolates. So, namely PQ2 and PQ3. So, our data have shown that the average mean of the validation runs is within the acceptable range from the predicted mean for the biomass yield. So it means that the optimization can be considered as valid.
Okay, thank you, Mr. Herrera. Yes, please, Mr. Vito, if you can approach the microphone.
I'm Lawrence Vito from PLB. I would like to ask a question for Mr. Herrera. Did you analyze the lipid analysis for your optimized isolates? And the second question is that refresh my memory, this is isolated from wastewater, but wastewater treatment. Did you find out if these isolates can bioremediate nutrients coming from the wastewater?
Again, thank you for that question. So, in my presentation, we use BCMS not to quantify but to qualitatively check for the presence of PUFA, EPA, and DHA. So we are just waiting for the for our what do you call it? For the standards, no, in the BCMS. So thank you. And for your second question.
For the second question, is it wastewater you isolated from the isolate of that particular organism? Did you try to study the bioremediation process of the particular isolate, or did you have any?
Okay. In our DOST Picard project, that is one of the objectives. No, but in my presented study, this is not the scope of the bioremediation. But based on the results of the pH and salinity characteristics of the isolated Trostochitrid, so we can use those data if we will be having another growth behavior experiments using the raw and sterile wastewater towards zero waste discharge. So that will be one of the goals of the project. Thank you very much. Thank you.
All right, let's move on. Yes, ma'am, please approach the mic.
Good afternoon. The question is sorry, I forgot
Miss Relucio San Diego.
Yeah, this is for curiosity, I guess. You sequence the whole genome, the two strains, right? From the where you collected your isolates. And so, and then we've done some prediction analysis to actually determine secondary metabolites, etc. So have you actually confirmed for those that you have detected in silicoid, they're actually, produced by this strains, and if you've done it, have you also quantified, if you have done some sequence or expression analysis for this?
So I just want to clarify. So we got the activated sludge, then we extracted the DNA from the sample. Kasi yung pinasequence namin. So it's a shotgun sequencing. So it's not, we did not isolate the microorganisms per se. We just performed the extraction. Tapos, when the sequences came back, we used assembly tools, then binning tools para mag-generate yung mags. So that's why nasabi namin na itong sequences na to belongs to this bin because of the algorithm of the mags. Of the bins, then we refine them. So actually, yung total na bins na na-generate namin from this study is 76. So, ganun siya kadami. And if I present yung lahat, hindi na full time. So, I just presented the best, if I may say, the best bin that we have. And we, yun present ko. So, as for the, titignan mo kung talagang siya na-produce, we did not do that kasi putative itong study because it is not. And mahirap, although may reports that it can be isolated, the data na species, hindi na namin na-focus.
Thank you. All right. I think the others can be presented in the 53rd PSM. All right. So, other questions from the floor, please? Yes, sir. Please approach the mic.
Good afternoon po. Kasi Paul from UPLB. My question is for Sunday. Since yung isolate meat from a storing environment, yes. So yung pH na leaning towards slightly alkaline, why use pH 1 to 7 range in your optimization studies, and why not follow up with yung sa pagumpang alkaline? For your question, we considered the range in our optimization runs the pH 1 to 7 because this is supported by several literatures wherein trostochitris can thrive in low pH conditions. Additionally, the other objectives from the project, we will be using raw and sterile wastewater. And then one of our game plans in our experiments, we will be changing the pH and the salinity of the wastewater, low pH and high salinity particularly. This is to inhibit the growth of unwanted of the microorganisms present in the raw and sterile wastewater, so that our isolate trostochitris can thrive in those conditions. So.
All right, thank you. More questions on the floor, please? We still have time to accommodate one question. Go ahead. Please approach the microphone and please state your name and affiliation.
So I'm Malan from UPM, Department of Bio. So first, congrats on your work. But I just wanted to ask, what's the percent similarity of the diseases that we were able to identify based on the tools that we use person and sa class.
Sorry, actually, yun nga, actually, hindi talaga siya. Yung percent similarity for the beta-lactam, hindi lumabas. For the rubiginone, it was less than 10. So, hindi pa rin siya talaga mas sustain na yun siya. Pero I have 2% na yun yung nilabas ni Ate Smash na analysis. So, we are, I'm not sure yet how we could look deeper na parang talaga bang hindi similar or do we have to do another sequencing just to be sure kasi meron kami bagong samples from the same sludge. So, mababa rin yung percent similarity.
Yeah. Okay, so for more understand kasi diba na-mention mo na we did like shotgun.
Yes, yes.
So perhaps hindi nga complete yung so yeah, so we yun yung medyo comment ko lang kasi actually ito yung confusing part with using those tools. Minsan there's a difference between BGCs and then the metabolite itself. It's possible na yung BGC hindi talaga yun yung actual na nandun siya. Kasi kung hindi ganun ka-complete at hindi ka sa similarity. But anyways, it's a good starting point, especially kung titignan niyo din yung novelty na, especially later on, kung makita niyo talaga na mababa pa rin yung percent similarity, pero yung scaffolds and all is almost similar to the BGC, then perhaps you might be able to get like double metabolism and all, which should be very interesting.
Yes, because yung, when I read some of the literatures, yun nga ginagawa nila. You repeat the sequencing again there, tapos i-compare mo ngayon yung first sequence mo sa second, lumabas pa rin ba siya? Or higher pa ba? So parang nag-increase yung confidence mo na nandun talaga siya.
Alright, so I think that is all the time that we have. I thank all the participants here for your thought-provoking questions and congratulations and a round of applause for our second batch presenters. And at this point, I would like to call again on stage our PSM President, Dr. Howell Ho, and our technical paper chair, Sir Emerson Cruz, to award the certificate of appreciation to our presenters. Let me read the citation. The Philippine Society for Microbiology, Inc. awards the certificate of appreciation to Mary Ann Cielo Lucio San Diego, titled Metagenome Assembled Genome of sludge of a poultry wastewater treatment plant reveals potentially high biosynthetic capabilities. During the 52nd Annual Convention and Scientific Meeting of the Philippine Society for Microbiology, Inc. in cooperation with Trinity University of Asia with the theme Microbiology for Unity and Prosperity through collaborations and industry linkages held at the Hilton Manila via hybrid from July 2021 to 2023. Given this 21st day of July 2023, signed by Mr. Emerson Cruz, the Chair of the Technical Session Committee, and Dr. Howell Ho, our PSM President. The same certificate is awarded to Zander Carl N. Herrera, NDJ Cat Lee, CMB Masakupan, VP Liga, and Elsie Villegas for presenting the paper and paper production of parang part ata talaga yung pagbintang test. Tinetest na aking ability to do. Trees isolated from selected mangrove forests in Batangas and Quezon provinces, signed by the same signatories given this 21st day of July 2023. The co-authors here, if you can come up on stage with the presenter.
All right, thank you very much again to all our presenters for this afternoon's technical session. Our lead paper, Dr. De Leon, Mr. Vitug, Ms. Balaga, Ms. Velicio San Diego, and Mr. Herrera. A round of applause again for all our presenters and their exciting research results. So thank you for being part of the 52nd PSN Annual Convention and Scientific Meeting Technical Session B, and for your commitment to advancing the field of microbiology. Let us continue to collaborate, learn, and make a positive impact to the scientific community. Maraming salamat po.
Thank you po, ma'am. Music lang mo. Anong music natin? Dito na lang, paulit-ulit na lang.
Good morning, everyone, and welcome to the technical sessions.
Yung USB.